The overall goal of this procedure is to use DNA in situ hybridization to map heterochromatic sequences to mitotic chromosomes. This is accomplished by first dissecting brains of third instar Drosophila larvae or another tissue of interest. The second step is to fix the tissue on a cover slip. Next, the tissue is squashed between the cover slip and a slide to prepare the chromosomes for hybridization. The final step is to hybridize short synthesized oligoprobes to the squashed tissue. Ultimately, fluorescence microscopy is used to show the localization of sequences complementary to the oligoprobes on the chromosomes.
The main advantage of this method over existing methods is its simplicity. Most other chromosome in situ hybridization methods involve washes with large amounts of formamide, which can be expensive and messy. Our method requires no formamide washes, making it a cheaper, cleaner, and faster technique.
Although this method provides a simple way to visibly map sequences on the somatic chromosomes of Drosophila brains, it can also be used to map sequences on other types of chromosomes from different tissues and in different organisms.
For each slide to be made, prepare 4 or 5 tissues, but not more. For brain dissections, begin with selecting wandering third instar larvae and collecting them in a drop of 1x PBS. Choose vials to collect from that are not overcrowded. Under the dissection scope, use ultrafine tweezers to grab